The broadcast is now starting. All attendees are in listen-only mode.
All right. Well, good morning. It is my distinct honor and pleasure to be able to introduce my good friend for the last 20-some years, who's come to us to talk today. I've known Alan. I think we both graduated, did our fellowships the same year, obviously in different places, but Alan originated from Pittsburgh. We have a tie there only because that's where I did my training. And Alan did his undergraduate work there, but then migrated to Chicago at Northwestern, and he's been a fixture in the Chicago community since then. Did his medical school, his training at Northwestern, did his fellowship with Rich Wyatt, and then went back onto the faculty for Northwestern. He's one of the most accomplished physician scientists in our field as far as cochlear implants go. And he's also been very dedicated to education for residents and medical students throughout his career. I'm happy to say, he told me last night, it's no secret, right, that he's just been elected to be on the RRC committee. And so it's a very big accomplishment, and we congratulate him. Alan, come on up. I'd like to have you start, and it's a pleasure.
Thank you, Peter. So once again, thank you for the invitation. I'm very honored to be here. It's always nice to get away from Chicago and come to New York and see what a real big city's like. So just by way of disclosure, I have no disclosures in regards to this talk. And as you're sitting there thinking, oh, great, we're going to have a basic science talk here at 7:30 in the morning, and it's going to be boring. Here's the good news. I'm an MD. I'm not a PhD. I have the good fortune of working with a very accomplished researcher by the name of Klaus Richter, who is the brains behind all of our cochlear implant research. If you've ever met Klaus, he's just an amazing fellow. As I always say, he speaks about 5 languages, one of them being calculus and another one being physics. So I'm breaking this down so we can see the more clinical aspects of what we've done here. Now, I have this slide, I added this early this morning. By way of disclosure, so last night after the Cubs won the World Series, my phone just exploded. Now, as Pete said, I'm from Pittsburgh. I am not a Cub fan. I root for the White Sox, but I'm still a Pirate fan. But I have this slide up here of Gene Durlacki. So hopefully most of you know who Gene Durlacki was. He was a very famous otologist. He was in Chicago, worked with George Shambo. And I had the good fortune when I started as I started on his faculty, to have Gene there as one of my senior attendings. And he became almost like my father. Gene was probably one of the most rabid Cub fans I know. In fact, when Gene passed away 12 years ago, I had the honor of writing a memorial for him. It was actually published in Otology and Neurotology. One little fact about Gene, when Gene was in college, he was at Northwestern, he got offered a minor league contract with the Cubs and almost took it, but his father said no and made him go into medical school. So he's probably the most avid Cub fan I know, and I know right now he's probably smiling down on the fact that the Cubs won. So I just wanted to add that and give a tribute to Gene Durlacki. So when we think about cochlear implants, I mean, we've really come a long, long way. I mean, if you consider the fact that, we first started knowing that we can electrically stimulate the cochlea in the late 50s, started with some single channel implants not soon after, and by the late 80s, we had multiple channel implants. It's really not that long of a time, and think how far we've come. I mean, we've really, a device has been developed that's been really, really beneficial to these patients. And I think it far exceeds what we expected for these to do. But just like anything else, we are high achievers as physicians and PhDs. We're always trying to make things better, stronger, faster, and that's, yes, for those of you that may have remembered the old television, so that's my $6 Million Man television show reference. So we know that we're using, our cochlear implants are electrically stimulation. So I have a couple of things I want to go through with this in this talk today. A couple of the things that we've done a lot of extensive research in our lab is, one, using infrared or optical stimulation. But the more interesting, and this is what we're going to focus on later in the talk, is this brain-to-brain interface. So we know that Dr. Doolittle could talk to the animals, but the question is, can the animals actually talk to us? So when we think about cochlear implant success, we're always trying to make things better. Because as you know, when you see these patients, while they think it's a remarkable device, they'll still tell you it's still not what I remember hearing. And of course, I'm talking about adult patients that are post-linguistically deafened. But they'll tell you that it doesn't sound like I thought it should. And music is never as good as it used to be. And what we typically tell the patients is that the device that you're wearing on the outside is a speech processor. It's not a music processor. But why can't we make it a music processor? So we've done things with making better electrodes, whether they were thinner, whether they were modular hugging, or we did hearing preservation electrodes, thinking, and as you know now, a lot of our implant patients have residual hearing, and fortunately we're able to save that quite often. A lot of the development, though, as far as improving the implants has been in the coding strategies. So can we refine this stimulation? In doing so, can we improve music perception? The other thing, too, is can we lose less energy to improve battery life? Now, I've always said that, we work with at least 3 implant companies that are approved in the United States, and I always said that the first one to get the totally implantable device that they can wear it all the time, don't have any external parts, that's checkmate for that company. They'll win. So, I mean, they're still driving toward that. I mean, Cochlear's had a sort of a prototype out there that they've used. The problem is so far is the microphone technology, and yes, there are still issues with battery life. So are there technologies we can come up with that can improve battery life? So we know that this high fidelity hearing that we have in a pristine system, a normal hearing ear, is really not restored. Now, like I said, I'm not taking this for granted. Our ophthalmology colleagues would love to have something similar to what we have with a cochlear implant. So some of the issues, though, with cochlear implants is that there's mass current spread. So you put the electrode in there. As you know, electrical current will follow the path of least resistance. So there's a lot of shunting of current up along the cochlea. So what happens is that you start stimulating larger populations of spiral ganglion neurons. Now, several years ago in our lab, and actually this was part of my trial thesis, we looked at resistivities, which is basically resistances across large pieces of tissue, composite tissue, in the cochlea, and what happens in a deaf individual. And what we found, so what we did is we took some animals and we deafened them, and then we were measuring the resistivities inside their cochlea. And what we found was that when an animal becomes deafened, the Rosenthal's canal, where you have all your spiral ganglion, actually becomes fluid-filled. So the resistivity actually drops. So believe it or not, a deafened ear system, fortuitously, became ideal for cochlear implant because actually most of the current's getting shunted into Rosenthal's canal to stimulate what we're targeting, which is the spiral ganglion cells. Some of the other things we've done, we've had multipolar electrodes, current steering, so shifting current between 2 adjacent electrodes to try to give different percepts in between those two. So like I said, this has been an ongoing evolving process. So once again, the question is, can we always do better? And the answer is, of course, yes. And like I said, a lot of this comes out of the mind of Klaus Richter. And this is not even thinking outside the box. This is some of the stuff that Klaus comes up with is sort of thinking outside of the universe. So when an implant patient uses a device, they need to understand and perceive these modulations to improve their speech perception. So a lot of these are temporal or timing sequences. So any condition that interferes with those aspects for modulation will restrict the information to the users. So because of that, we have several limitations within the devices. So most processors, when you pull them out of the box, they only stimulate between 250 and 2,000 pulses per second. Now you would think, geez, if we can improve that, they would hear better, but they have done some studies with that, and it really has not been proven that that improves one's perception with a cochlear implant. So Klaus sort of got this bright idea. Well, what if we use a different type of stimulation? You know, we know that electrical works, but we have issues with it because of current spread and so forth. And there was a lot of literature showing about using optical stimulation, mainly in the peripheral nerves. So Vanderbilt had a whole project that they were working on stimulating the sciatic nerve with basically infrared stimulation. So Klaus came up with this idea, well, can we hear the light? So can we stick an optical source in the cochlea and give someone perception? And there's some theoretical benefits to that. So first and foremost, what we're talking about here with the cochlea is what we call infrared stimulation. There's another type of stimulation that you see out there, they use it sometimes in deep brain stimulation, is optogenetics. So optogenetics works with the activation of photosensitive channels within the cell membrane. The problem with that, with the ear, is as follows, is that one, some of those cells may need genetic modification, and you do need direct contact. So one of the benefits of what we're trying to do is avoid some of that direct contact, because that will increase your specificity. And then the maximum stimulation rate with that is only about 70 hertz. In the cochlea, what we were using was infrared neurostimulation. And what it uses actually is it actually heats the target structure, therefore causing the firing. Now, with the optical stimulation, you do have finer spatial resolution. And like I said, no direct contacts necessary. The one drawback is that the rate of stimulation is limited by the heat dissipation. So you have to wait for the tissue to cool before you repeatedly stimulate it. So what we did was we had actually it was Dave Ryugo's colony of deaf white cats. And that's what we did all these experiments in, as far as looking at the safety of this long-term stimulation. And I'm sorry I don't have any of the pictures, but literally we created these little backpacks for the cats with the little laser, small laser on the back with these fibers going into their ear. And we stimulated them for a long period of time. And we found that at about 200 hertz, we could safely stimulate it for a long period of time without causing any cochlear damage. Granted, like I said, if you got any higher than that, we did start seeing some heat damage. So the idea was, with the optical stimulation, does the temporal pattern that we measure at the level of the inferior colliculus, does it differ using an acoustic stimulation versus the infrared neurostimulation? So what we did is we did an experiment where we were measuring responses to both of these stimuli at the level of the inferior colliculus. So it was a very straightforward experiment. We had about 12 animals, 4 normal hearing, 4 acutely deaf, and 4 long-term deaf. Did a cochleostomy at the basal turn, and then we placed the optical fiber inside the cochlea. Now this was the harder part. So craniotomy was done, and using a microdrive system, we sterilely tactically placed a tungsten electrode into the inferior colliculus. Of course, that was connected to an amplifier. All the experiments, we did make sure post-test that we did histologic specimens to confirm the placement. We did some with single channel, some with multi-channel, and they actually confirmed placement and responses to sort of low hertz tone pips. Busy slide. But basically, what you see mainly down here with the repetition rate is you see that the acoustic stimulation in dark was fairly similar to the infrared neurostimulation. So there really wasn't much of a difference. And what the measurable difference was that at higher rates of stimulation, it was pretty much the same. However, the phase locking that's necessary for a person to understand actually occurred at lower rates in the infrared stimulation. So there's an energy saver. It does have a little longer latency though. Now, there still are some issues with this. One is, like I said, the speed of stimulation is restricted by the heating issue. The other thing we found is that light can penetrate. So what we were early on when we first started this, we were getting stimulation of different target cells because we would stimulate one area of the cochlea, but the light would actually pass through and hit what's left on another turn. So even though it's very specific, the depth of penetration is greater. So it's not as specific as we thought. So, this whole when we first started doing this and a bunch of other groups jumped on the bandwagon with this, we thought, hey, this could be the next great thing. But it turns out that it may actually have a more ideal application as a hybrid device where you have electrical and optical stimulation. So you can fill in some specificity with the light, but you really still need the electrical stimulation. So we haven't replaced that just yet. Taking that information, we still thought, what else can we do to make this better? So we went back to the proverbial drawing board. So let me digress a second. So we have some speech processing strategies that are basically used by the current manufacturers. So there's ACE, which is Advanced Combination Encoder. There's fine structure processing, and there's a high-res 120 strategy. When a speech processor initially picks up a signal, using filter bands, the audio signal is broken down into these bands. There's an envelope of temporal and spectral information that's extracted, and that's used to modulate the carrier rate of the speech processor. Then that's put back into the patient in specific tonotopic areas. So that's where the audiologist sits there and figures out where this is best heard by the patient when they do their initial stims and their subsequent checkups. So the primary code information that we really need to hear with an implant is in the temporal envelope, so in this timing envelope. So the temporal envelope is predominant in the speech coding. What we're finding though is we really need to get some of this temporal fine structure if we want to improve upon that, because that tends to be more beneficial in noise, which everyone who has hearing loss complains about. It can't hear in noise, and also for the perception of music. Once again, we're faced with these following issues, that high current rates can lead to current spread. So the physiological characteristics of the scala tympani and the bony structures, I told you earlier, causes current shunting along the tunnels. And like I said, this can lead to confounding neural populations being stimulated. So when you really think about it, we can really only focus in on about 4 or 8 electrodes in the cochlear implant out of the 16 or 20, depending on what device you're using, that can seriously and function independently. There's also a very limited dynamic range in the cochlear implant users. It's only about 6 to 30 dB compared to about 120 decibels in a normal hearing person. So, once again, thinking, can we overcome these limitations? So a thought came out in our lab, why can't we start with a natural sound processor? So we had all these measurements to different words from the inferior cochleus on these guinea pigs. And Klaus came up with the bright idea to take that signal, recreate it, and say, maybe we can present this back through the implant and see if these patients can hear it. So basically what we're doing, the thought was take a normal hearing cochlea in a mammal, use that as the initial speech encoder. So use that instead of a mathematical equation to extract these speech bands that we initially break it down into, and use a cochlea to do that for us. Feed that back. So basically what we're doing is we're trying to save some time and we're trying to put more information. So basically, if we take a measurement from the inferior colliculus, which has coding information from the cochlea and the eighth nerve and some of the other structures as you go up the brainstem, if we feed that information back into the cochlea, can a person hear it? So now you're taking naturally coded information. Since you've already had some of this information decoded for you, that's going to lose less energy. You're going to decrease the population that you need to stimulate. And what this will allow, theoretically, is for more individual channel stimulation. So we can be more targeted using this signal, and therefore we can actually take advantage of more of the electrodes without getting channel-channel interaction. So, very simple experiment. Haha. Basically what we did is we took CNC words that you were typically used in your hint testing, and these were played to anesthetized guinea pigs. We were taking single neuron measurements. We used about 100 distinct neural units corresponding to the frequencies up here from about 500 to 16,000 hertz. Once we took these recorded audio files, we converted these into a vector containing primarily the spike timing information. And then, once we had this information, we tried to correspond this to the closest electrode to stimulate in an implant user based on tonotopicity. So basically, take the word bomb. This is one of the words we used, and this is sort of the spectral frequency of that spoken word. This was the neuronal response at the level of the inferior colliculus. So what we did is we took this information and generated a spike train that went over, about, between 0.2 and 0.5 milliseconds. So this is what we generated. This then was fed back into a cochlear implant patient. So basically we had about 12 patients, 6 men, 6 women. All of these were advanced bionics user because what we were using was their research system, one being the bionic ear data collecting system, the BDEX. We used that on all 12, and then 5 of them, they had an upgraded model, which is the HR stream. So basically, this is just using this system instead of their speech processor to feed this information back into their implant. So we established comfort levels for the listening for each patient. This is just the demographics of the patients. You see the youngest was about 47, oldest was 85. Like I said, they were all advanced bionics users. All of them were tested with the BDEX system, and then 5 of them used the HR stream, and we'll talk about there's a little difference between the two. So after we recorded all this, we basically chose 21 words. And we were able to use about 6 to 12 channels depending on the word that we were using and depending on the patient. In the first experiment, we actually did do a training session with the patients to give them information about the words they would be hearing. After that, we did, in the first trial, then the testing was done with no further instruction, and we simply asked the patients, what did you hear? We then brought them back with no further training. We had a second and third training, asked them just to describe the words, and then we did about 4 to 10 sessions giving them forced choice. So they basically played it to them, gave them a choice of 4 words, and see if they could pick out what they thought they heard. So the very first 2 subjects we did, we got very excited about this because they were able to discern the length and length, rhythm, and loudness between different words. So they could hear something on a lot of them, and they would say that it sounds like speech. And they were also able to pick up rhythm and timing differences with these signals. Sometimes the subjects were very wrong with what they were saying, but they were also very consistent. One of the funniest was the word ditch was played to them, and Klaus, being from Germany, she kept saying, she goes, what do you hear? And he goes, Wilhelm. And he kept saying, what? And she goes, I hear Wilhelm. So Klaus actually thought that was funny. She thought thought she didn't know his name. But they basically did hear this signal that we pulled out of the inferior colliculus of a guinea pig as a word. When we took it to the forced choice results, when they had the four words to choose from, in the first trial, the group on average got about 33% correct. Second trial, they got about 31% correct. But if you look at overall the number of words that they were actually hear, that group was actually able to hear a little over 50% of those words. So they were actually to say that this signal we pulled out of the inferior colliculus was the word ditch or was the word bomb. So they could hear a little over half of the words very clearly. So this is rather interesting and rather exciting. So this is just, each subject's testing. It was curious that they did better on test one. Don't know, real explanation for that. But like I said, though, the overall recognition between the patients that they were able, between the two tests, to hear a little over 50% of the words. So this is just another scaled look at this. And then this is the estimated probability then. So what we're thinking is, like I said, using this type of stimulation, we were able to get about 30 to 40% of the words correct. But if you, over a period of time, these patients may have the possibility of recognizing about half of them. So what does this all mean? We definitely saw that the patients did better when they had training. Now, while this was not open set because they were sitting there with 4 words to choose from, it's pretty remarkable to think that we took a signal from an animal, an electrical signal, put it back into a human. So you have a signal that's been pre-processed, and they were able to hear it. So there is a low level of speech perception that is occurring. So what this tells us is that between the cochlea and the inferior colliculus in a mammal, there is some coating of temporal fine structure, and there is some coating of the temporal envelope. And that's really important to know. So by presenting this data, we're taking away, actually we're sort of cutting back the work that the cochlea, the eighth nerve, has to do for these patients. Now, there were some issues with this. So the BDEX system that Advanced Bionics first gave us really only uses a small number of electrodes. When we upgraded to what they call the HR stream, it did show the results trended to be better because we're actually able to incorporate more electrodes into this. Obviously, training is important, and we do that with all of our implant patients anyway. The question is, though, can we improve? One of the thoughts was that we probably need to improve on the signal we are extracting from the animal, because when you put these micro drive systems and you target these things, and you're trying, especially if you're trying to get all these different placements in the inferior colliculus, you're not always going to be exactly on target where you need to be. So finding a guinea pig where you get a perfect reading in the perfect place is a little bit difficult. But like I said, it's still pretty fascinating we're able to do this. So what does this hold? So coding strategies still really rely more on place theory as opposed to volley. Volley means you can stimulate multiple different areas at once. The problem is that this ignores phase locking. But what we think, though, is that by using a functioning cochlea initial process, it avoids some of these issues by naturally encoding the sound. So like I said, all the encoding of sound that happens with a speech processor in the cochlear implant is all mathematical. So here we are taking something done by another mammal and feeding it back. So we're using that mammal to do some of the math and some of the filtering for us. So what are some of the future benefits? We are able to increase loudness by increasing the pulse rate. The other thing is we don't have to modulate the amplitude too much because the signal has been pre-processed. Finally, there is a constant current level. So a lot of how we modulate things is increasing, decreasing current. If we can take this pre-processed sound with a decreased current, this is our future drive into getting a totally implantable device. So what this may do is lead to an increased dynamic range in these implant patients. Because like I said, our dynamic range is pretty small in implant. It's only up to about 30 dB as opposed to a normal hearing error, which is about 120. So while we have an increased rate with this, using this type of information, it's still less than our current devices. So if we can use less current, this limits current spread. So if we can eliminate current spread and electrode-electrode interaction, we therefore can lead to more functionally independent channels. What's this lead to? This leads to longer battery life. And like I said, the ultimate goal eventually is to get maybe a totally implantable device that's on 24/7. So this also, by using this type of strategy, may be compatible with using more of the light stimulation, the infrared nuclear stimulation. So the future benefits could be perhaps better hearing and noise. I mean, patients will still tell you that's a big issue. The bigger issue, especially if you have some, get these older implant patients that really have an appreciation for fine music, they'll tell you that it's just not the same. So if we're able to take advantage of some of this pre-processing that's done by the animal, can we therefore improve what we're stimulating to humans, use less energy, and overall improve the quality of sound for our implant users? Like I said, I give great credit to Klaus. Klaus is our lead researcher in our labs. He's sort of the brains behind this. And then the rest of some of the other people in our lab, my audiologist, Pam Fiebig, who helped with some of the trials we ran here. That's all I have. Yes, sir.
So it's probably a naive question, but how do you know that because you're using the experimental cochlea as a speech processor in a way, or a speech coder?
Correct. You're using the animal cochlea to pre-process the sound.
And how do you know that that's appropriate to code for human sound?
You don't.
Right.
So that was the reach.
How do you know that little animal isn't hearing Wilhelm
Instead of hearing,
that's right,
and hearing, yeah.
is red to all viewers of a rose?
You don't. So the question is, if we're, how do we know that when we do play a word to the animal, that it's not really hearing what the person thought they were hearing, and you don't. But the fact that some of the responses that the patients received were they could pick up some timing, but they really couldn't say the word. But the ones that they did hear fairly clearly were quite remarkable. So, but you're correct. You don't know if that code and that signal, that envelope that we created to feed back into that experimental implant system really isn't encoding Wilhelm instead of encoding.
If they were all hearing something different, then you would say, well, that's not necessarily, but if they're all hearing the same word in certain circumstances. But
that was, so that the Wilhelm was just funny because Klaus is German, but it was only the one patient was able to do that.
I see.
But you're correct in that those recordings, while we did histologic placement, and when we took that envelope that we extracted from the inferior colliculus, even with a microdrive system, you're still not absolutely perfect in where you're thinking you're getting this. So is that a consistent signal? And the answer, that's still up for question.
Right. And then the other thing is you're switching out from if I understand this correctly, amplitude for frequency.
So you're able to. So the thought is, we're able to add more temporal coding.
Right.
And so when temporal coding does improve our perception, as we know, and by using temporal instead of this amplitude and frequency, you're decreasing the energy that's needed to stimulate the system.
Right. And is that more biological? shift?
And the shift in what?
Well, you're going to a lower energy system, and is that more akin to what we use in our brains to process words and hearing?
Not necessarily, but if you can, the problem is that the reason we're trying to decrease the energy that's required and actually decrease the placement is what's left in the survival of the cochlea. So if we can take some of this higher frequency information, extract this code and replay it at a lower amplitude, lower frequency, where we typically have more survival in the lower frequencies, it's more beneficial. So what we're doing is sort of taking higher frequency and having lower frequency neurons hear this. And so that's what we're trying to do to take advantage of what we have left to work with in this system.
How are your animals deafened?
Neomycin.
Neomycin. So I'm just curious if you deafened the animals by different methods, if you got the same brainstem recordings.
Good question. So the question was, if you deafen them by different methods, one drug versus another drug versus sound, noise, and that's a good question.
So 2 questions. The first one, of the patients that heard words, those 33%, 50%, were they consistent in the sense that everybody heard whatever it is, ball or ditch or whatever, they heard those words, and consistently most of the patients then could hear the same words.
Correct. So the words that there was a 33%, 37% correct in the first, and 31, 32. They were pretty much the same words. But like I said, there was not a statistically significant number, but there were enough words that at one point, this group of patients could hear somewhat reliably about 50% of the words from trial to trial.
But were they the same words?
About a third of the words were the same.
Yeah. So that's what I mean, is that they're, back to Eric's question is, are they hearing the exact same word we are? 30% of the time it sounds like maybe they are, which is pretty interesting.
It is interesting, and the interesting thing too is like I said with the Wilhelm example, because we got a good laugh out of that. It was consistent though.
Yeah.
We let her go and do. It was a woman. We let her do some other stuff. We played back that word again later on in the trial, Wilhelm. I mean, that's what, and,
So Gary Larson may be right. What dogs hear.
Yes. Well, and the question too is, when we're taking these electrical recordings out of the, inferior colliculus, we're probably not 100% exact.
Yeah.
But like I said too, do we really know, how much different the processing is between the cochlea and the inferior colliculus in a gerbil versus it is in a human?
Were you able to look at any? The other question I had is, in my mind, I've looked at, how can we do clarity better? And if you look at a speaker system, you have a bass, but the bass, that apical end, that's just sort of the icing on the cake, but you need it in order to create the whole sound. And as you intimated, we tend to really stimulate more on the, sort of basal high frequency. Did you look in your methods of just maybe tweaking that and stimulating not the whole apical end as you were talking, but just, maybe one little part of it to fine tune it?
Well, like I said, when we mapped these things out, we did try to use as many electrodes as we could, because one of the hopes is that because you had a pre-processed signal that you're feeding into them that was already more fine-tuned, and we were hopefully able to use less energy, we could spread the stimulation out amongst or between all the electrodes,
yeah,
to increase well, to increase the spread, I mean, increase sort of the coverage, but since we're using lower energy, there's less spread. So the idea is that this avoids or at least reduces some of the channel-to-channel interaction. So this, like I said, we tried these stimuli along different channels along the cochlea, seeing obviously where they heard it best. And what we did find though is we were able to independently stimulate up to 12 electrodes in some people, because it did use much less energy.
Super. Thank you very much.
Thank you very much. Nice job. Thanks.
Yeah.